this last section is about what happens when there are changes to the DNA. And those are called mutations. So changes to the DNA can happen just by random errors when DNA makes copies of itself, although it has enzymes that have the specific job of looking for mutations and problems and fixing them. Sometimes that can get overlooked. And there are also chemicals and physical exposures to things that can increase your chances of mutations. For example, ultraviolet light can cause mutations in DNA, which is a lot of times how skin cancer comes about. So there's a lot of different kinds of mutations. What we're going to focus on actually in this chapter is what are called point mutations. And that has to do with substituting, inserting, and deleting nucleotides. Meaning, like, an extra letter is added or deleted, or the wrong letter is in a specific spot. So that's what we're talking about. Sometimes a mutation has no effect at all, because there are areas of your DNA that aren't even copied. Or, remember how when we looked at the genetic codon chart, some of the codes coded for the same thing. So if, like, ACC got changed to ACG, it might still make the same amino acid in the end, and that mutation would have no effect at all. But pretty much all genetic disorders are mutations in a gene that result in either no protein being made or the wrong protein being made. So the protein has the wrong shape and behaves differently than it should. For example, with sickle cell anemia, the protein hemoglobin is made incorrectly because of a mutation in the DNA, and then it folds up wrong, and so the blood cells are shaped wrong, and then you have all these symptoms. If you're lactose intolerant, maybe you have a genetic mutation where that protein enzyme, lactase, that digests lactose, is not made at all. And so that's the result, is that you then cannot drink milk without getting sick. So genetic disorders are typically the result of mutations in the DNA. Now, we've talked about already chromosomal mutations. This was actually discussed in another chapter, like where an entire section of a chromosome is missing, that's called a deletion. Or if a part is copied twice, that's called a duplication. So you're not going to be tested on chromosomal mutations in this chapter. We're going to focus only on gene mutations. So just a mutation, a small mutation that only affects a single gene. Because if you're missing a whole section of a chromosome, there might be 30 or 40 or more genes that are affected by that. But if it's just one gene, it might be minor. It depends what that gene codes for, right? If it's a gene that codes for some protein you need to live, then it's deadly. If it's a gene that just codes for, like I said, digesting lactose, okay, so you can't drink milk. It's not the end of the world. So, anytime a mutation just involves one or two, just a couple of letters of DNA, oftentimes just one letter, it's called a point mutation. And the three we're going to talk about are substitutions, insertions, and deletions. So, a substitution is where there's a single letter changed in the code and the wrong letter is in that spot. So it's substituted, right? So if we take a look at this example here, this is the correct code. This is the DNA code, yes, and this is correct, and it's supposed to have a C here, which would then, in the mRNA, code for a G, and then this codon codes for arginine. But in this substitution, there's a mistake in the DNA. Instead of a C, it has a T here. The mRNA now makes an A instead of making a G, and this codes for a different amino acid, histidine. Now, what does that mean? Well, it could mean that this protein now folds up the wrong way, because once this protein is made, it's going to have a specific job. And if it's an enzyme, if you remember, enzymes have a special active site for a job. For the example of hemoglobin that I gave you, your red blood cells are supposed to be round and kind of dented in the middle because of hemoglobin. In the case of sickle cell anemia, it's literally one amino acid that's wrong out of something like 286 amino acids in the chain, I want to say. It's a really big number. One amino acid is wrong because of one letter in the DNA that's wrong, and the entire red blood cell folds up like this into this pointy sickle shape, which doesn't carry oxygen well and causes all kinds of problems. So, even though it only affects one amino acid, unless this changes to a stop codon, then of course the whole protein would terminate. This could still be a big problem. I mean, it could be minor, it could be big. It really just depends how important that amino acid is to how the protein folds up into its final shape and what its job is going to be. But insertions and deletions are typically a bigger problem because you're going to affect more than just one amino acid. You're going to cause what's called a shifting in the codons, and it's called a frame shift mutation. Now let me show you why. Remember how the DNA reads in it's read in 3-letter codes. This forms your 3-letter codons. Okay, if you insert an extra letter here, so now there's a T here, what's going to happen is this C is not going to be the 3rd letter anymore, right? Now it's TAT. This C is going to be part of the next sequence. Now it may not be as easy to understand here in this picture, but we made a sentence here of 3-letter words to kind of show you what this means. But in other words, the reading frame is shifted. So instead of reading AUG and then CGU, this gets shifted over. So now, because of this extra T, we're reading CGC, ATG, GAA. In other words, all of this has been moved over a letter. So the reading frame has shifted. Now, you might say, I still don't understand how this causes a problem. Well, every single one of these codes is basically wrong. But take a look at this sentence. I think this really explains it. The fat cat ate the rat. If I delete the H, but I still read it 3 letters at a time, look what happens. Now the F from fat becomes the 3rd letter here. And now this is my 2nd set, this is my 3rd set. Every single word is completely wrong. If I add or delete, I'm going to shift my reading frame over, and in this case, that means it's going to change these 3-letter codes, all of them, not just one. Remember, a substitution just changed one amino acid got changed. In this case, everything after the point of the insertion or the deletion is going to be incorrect. So this is typically going to be a much, much bigger problem. Probably you're going to make a non-functional protein when it comes to a frame shift mutation. All right, sickle cell is an example. I was talking about it before. Normal hemoglobin in this particular spot, the DNA has this code. The normal hemoglobin has glutamine. In a mutant hemoglobin, the gene for sickle cell basically has the wrong code here. It has an A instead of having a T, which means this becomes GUA, and this valine is the amino acid that's substituted. Now that probably doesn't sound like it means a whole lot to you, but it turns out that the properties of glutamine and valine are extremely different. So the way these interact with the rest of the chain and cause it to fold up change the entire shape of the red blood cell, and it becomes a major problem. All right, and that's actually our last part of the chapter. So hopefully this was informative and helps you for the test.